Tina Sawyer.
And I'm Brittany Souza.
And the topic we chose was about the Ebola virus. 2014 was known to be the greatest epidemic of the virus.
The article that we found was called the Chimpanzee Adenovirus Vector Ebola Vaccine Preliminary Report. This article further explains vaccinations that were created to prevent the outbreak of the virus. A small study was done on 20 participants to test the adverse effects and the effectiveness of the vaccination.
The reason the study was performed was because the scientists are trying to find a vaccination to help the people. Ebola has been around since 1976, but has recently been more known because of the current outbreak of the virus and how many people have been affected by it. Scientists have discovered that of the 5 known viral strands, 2 of them, Zaire and Sudan, were found to be the strands to cause the most recent Ebola virus outbreak.
2014 was the year that had the largest epidemic of the Ebola virus. In July of 2014, over 650 people in the United States were showing signs of being infected with the virus. 61 people were tested and 4 were infected. Since the outbreak of the Ebola virus, appropriate precautions have been taken place. Protocols are enforced, such as monitoring programs to monitor symptoms that may be an early sign of the virus. The 2014 outbreak has also initiated an international response for the development of a preventative vaccine. A replication defective recombinant chimpanzee adenovirus type 3 vectored Ebola virus vaccine has been created with the glycoproteins of the Zaire and the Sudan viruses. The vaccines were created and first tested on a non-human primate. The vaccine protected the subject and advanced to the next stage of creating the
study to find an effective vaccine that will prevent the Ebola virus. This study was used to test the safety and efficacy of the vaccine that is developed to protect against the Ebola virus infection. Once the vaccinations were made and tested, a group of participants were asked to participate in the testing of the effectiveness of the vaccine. Participants were divided into 2 groups. 10 were given the Zaire vaccination and the other 10 participants were given the Sudan vaccination. This study has been presented to determine if the vaccine induces an immune response to the virus. Participants were from Washington, D.C. The study was conducted at the National Institute of Health. All of the participants were required to give informed consent of the study trial. The vaccine used was a sterile aqueous solution that contained glycoproteins from both the Zaire and Sudan species. These 2 species were used because they were known to be the causative factors of the Ebola outbreaks. The vaccine was administered via intramuscularly. There was safety monitoring that was being conducted, as well as laboratory and evaluation tests during the first technique conducted was the Ebola glycoprotein-specific antibody responses. The glycoprotein titers were evaluated with an ELISA test. If there was a positive response, then there was a significant increase of concentration in the titer. These tests were performed to measure both species at weeks 0, 2, and 4 of the trial.
The second technique used was the Ebola glycoprotein T-cell responses. T-cells were assessed with a peripheral blood smear at weeks 0, 2, and 4. These were evaluated to determine the proportion of CD4 and CD8 T-cells. These participants were considered positive if they had a response to either CD4 or CD8 at the weeks of 2
The third technique was a CAD3 and an AD5 serological assessment. This assessment was performed to see neutralizing antibody titers. It was also to determine the differences of the baseline and the vaccine-induced neutralizations of CAD3 and human AD5. According to the participants' responses during the visits, the reactogenicity of the vaccine was mild to moderate. Medications were strictly used for symptomatic treatment, such as lowering body temperature. Fever was reported and developed 8 to 24 hours after initial vaccination, but these symptoms were relieved with antipyretic medications. As for the Ebola-specific antibody response, at 4 weeks, the anti-glycoprotein antibodies for the Zaire species were found in 90% of Group 1 and 100% in Group 2. All 20 participants had an anti-glycoprotein antibody response to one species or strain. For the Ebola-specific T-cell responses, 20% of the participants of Group 1 had a glycoprotein-specific response to CD8, and Group 2 had 70% participants show glycoprotein-specific responses to CD4. The CD4 and CD8 T-cell memory was greater in week 4 than in week 2 of the study. The CAD3 and AD5 serological assessment reported that any background immunity can alter the effect of the response to the virus vector.
The results of the study report that the vaccines made are safe for use. All of the clinical manifestations of the study have had a relation to previous studies of other adenovirus vectors. The Zaire species is responsible for the greatest epidemic in 2014. The Zaire vaccine has been prepared and has shown great efficacy. The United States continues to review the vaccine through various clinical trials and will hopefully be approved to stop the outbreak. For now, all we could do is take the appropriate precautions by using active monitoring programs and safeguard protocols to save people in the United States.